Good evening, everyone, and welcome to our event for tonight. This is our July edition of the Caltech Astronomy Public Outreach Lectures that we host once a month. I'm Dr. Cameron Hummels. I will be the MC for tonight's event, so thanks for joining us. The schedule for tonight, after a few brief announcements by me, I will introduce our speaker for tonight, who will talk for about half an hour. She's going to talk about life in the universe and ways of detecting it, and so on and so forth. And then we'll immediately go into a Q&A session consisting of 5 different members of the Caltech Astronomy, Astrophysics, and Planetary Sciences Department to respond to questions from all of you on questions related to the topic of the talk, or related to astronomy or physics or space science or planetary science or what have you. We will do our best to respond. So these events happen once a month. We normally, obviously, we do them in person at Caltech itself, but due to the pandemic, we've moved these fully virtually online since March. And our next one will be August 7th, I forget, something like that. It's the first Friday, I believe, in August. And we'll announce all of the things on our website as well as on our YouTube page. We also accompany these events. There are astronomy on tap events that occur once a month as well. These are slightly more informal events that typically take place at a bar. Of course, again, we're not doing them at a bar right now, but we are continuing to hold these. The next one will be a week from Monday at 7:30 p.m. And it's all about Mars and the Mars 2020 Perseverance mission that's coming up. It was actually supposed to, the launch for Mars 2020 was supposed to be a week from Monday on the very day that we were doing it. And we have members of one of the instrument teams and another recent PhD graduate from our department talking about Mars and how to, what we understand about it and how to explore the surface of Mars from your own computer. Like there are a variety of different tools that you can play with online to be able to to go over the surface and explore the features of Mars that anyone can use. So that will be a week from Monday, and then there's pub trivia afterwards. And because it's meant to be in a bar, most of us, and maybe you as well, will be consuming alcohol. So you're welcome to join us for that. On our YouTube channel, I've already made the event, so you can already set a reminder and check if the time works for your schedule or not. I think those are the main announcements. Normally, these events have a stargazing component, which of course is made difficult by being online and being stuck in my apartment. But last month we talked about what was in the sky. So if you're here in Pasadena or in California or, well, lots of places, the things in the sky visible at about, well, right now nothing's visible except the sun because it's very bright out. The sun doesn't set until about 8:15 here. But in the early evening after the sun has set, starting this month, the most interesting stuff are Jupiter and Saturn are visible. They should be very obviously visible. They will look like very, very bright stars. But if you were to zoom in on them with binoculars or telescopes, you'd see the angular size to them. And even with small telescopes, you can see the red spot of Jupiter or the rings of Saturn or the striation and the different bands on Jupiter. So that's really cool. And the other highlight in the night sky right now is a new comet, kind of the brightest comet that we've had in around 20 years or so. It's called Comet Neowise. It's mostly only visible right now in the early morning, just before sunrise. So if you were to get up at like 4 or 4:30 and look in the northeastern part of the sky, kind of towards where the sun is going to be rising, you'll see, you can make out this band of the comet, and we'll continue to see it for most of the month of July, presumably. but in the next week or so, it'll shift where instead of being up early in the morning, it'll be up later in the evening, which will be better for night owls like me, because I don't want to get up at 4 in the morning to go watch this comet, although I probably will. So anyway, that's another really cool thing to check out, and I encourage people to, especially if you're an early riser, to maybe peek your head out before the sun is up and see if you can catch a glimpse of Comet Neowise. Okay, so I think those are all the announcements for me. I would like to introduce our speaker. So our speaker tonight, go ahead and pop on, Cecilia. Cecilia Sanders is a PhD candidate in geobiology at Caltech, where she studies the geologic record for information about life forms on ancient Earth. This is an extension of her previous work as a master's student of planetary sciences when she was intrigued by the preservation problem. How do living things impact the world around them in a way that is preserved in rocks for millions to billions of years? Cecilia tries to answer these questions by measuring and sampling assemblages of ancient sedimentary rock and by creating models of the ancient world using living organisms in the laboratory. Her work has taken her to Brazil, Namibia, the Turks and Caicos, all over the southwestern United States, and even back home to the fossiliferous cliffs of Maryland and DC. And when she isn't doing research, she teaches science to kindergarten, first and second graders in Pasadena Unified School District, and is a community activist for racial justice. So, welcome, Cecilia. Do you want to go ahead and share your screen? And I'd also like to point out that Cecilia is an amazing artist, and many of the slides that she's going to show, she actually did the artwork herself, which I think is incredible. So I'm really looking forward to this talk.
Awesome. Thank you so much, Cameron. I'll go ahead and share my screen.
Awesome.
All right. How's that looking?
That looks great.
Amazing. All right, then. So thank you, Cameron. Thank you, Caltech Astronomy, for having me today. Let's dive right in. So as Cameron said, my name is Cecilia Sanders, and you may have caught that he mentioned that I'm a PhD student in geobiology at Caltech. And though I do have my master's in planetary science and an undergraduate degree in planetary science and astrophysics, I am not an astrophysicist, and I am not an astronomer like most of the other speakers in this talk series. Rather, I study the history of life on Earth, and my thesis is about carbonate rocks in eastern Brazil and bacteria. In fact, the background that I have for you today is a photograph from the top of Morro do Pai Inácio, please excuse my Portuguese pronunciation, in Brazil. So I'm not necessarily the person you'd immediately think to go to with questions about space. But my journey as a scientist started with astronomy, because when I was younger, I wanted to know the answers to questions like, are we, the living creatures on Earth, alone in the universe? How did life begin? Why is life the way that it is? Why is it where it is in the universe? And also, what is life anyway? And it seemed to me as a young person that in order to answer these questions satisfactorily, then you would have to go all the way back, not just the first human, not just the first plant or animal, not the first cell, but back to the geology, the structures, the chemistry of planets and the stars that birthed them. And so I started my journey to science, zoomed way, way out. And over the course of years, I've moved back in and looked at the planet that we live on, because as far as we know, Earth is the only world with life on it. And if you want to understand life, then you really have to understand the Earth. But ultimately, my underlying curiosity, my motivator, has always been the same. How do we search for life on other worlds? What are you actually looking for when you're looking for life? And it's such a simple and incisive question. It's a child's question. The question, Mama, what is life? And really the best answer I've ever heard is, well, baby, you'll know it when you see it. Let's see if I can advance my slides. Whoop. Right, so this answer, you'll know it when you see it, is a little bit glib, but my hope is that in the time we have together today, I can try to expand on that a little bit and try addressing how scientists define, describe, and detect and recognize life when they see it in the remains of ancient Earth or on other worlds like Mars or Europa, or planets beyond our own solar system. So I know that I'm not alone in my fascination with life, what it is, where it came from, what it's doing in the universe. You just have to look at most examples of popular science fiction, Europa Report, Star Trek, Contact. There are any number of science fiction novels, films, and television series, radio plays, and indeed much of the real world space exploration agencies and organizations. All of these characters, all of these people, we are all motivated by the search for life and the study of life in the universe, at least in part. But what are these television characters and what are modern scientists in our world actually looking for in the first place? And how do they know they've detected life signs unless a tentacle jumps out of the water and literally grabs them? What are we actually looking for out there? And how do we distinguish between a non-living thing like a rock and living things, things that we recognize as living things like microorganisms or microbes? What is it that makes rocks, clouds, oceans, suns, and other things like this different from the living part of our world? First and foremost, I'm sure that you've heard whenever there's a announcement of a new habitable planet discovered or possible evidence for life in the universe, usually the first thing that you hear people talk about is water. You need water and you need rocks, or at least a rocky planet, a solid planet. The reason for this is that rocky worlds tend to have, well, in addition to just a surface to walk around on, they can achieve the right temperature and size and other properties necessary to have liquid water at the surface. Rock itself can be a source of this water, and it can be a source of other important chemical compounds that can help fill the oceans with useful molecules during the planet's early evolution. And water is particularly essential because lots of things are soluble in water, lots of these useful chemical compounds, these useful molecules. Water provides a nice stable medium for movement and the exchange of molecules across membranes. But despite this essentialness of rock and water, apparently, to our definition of life in the universe, I think that we can all agree that water, rocks, and sediment, and a bunch of random molecules just floating around in it don't really add up to life on their own. It's not life as we define it or recognize it. So perhaps it matters to us, matters to our definition of life, what kinds of molecules are actually floating around in that water. Because as it turns out, there's a difference between organic and inorganic substances, the substances that make up the structures of living things, and the substances that make up a lot of the non-living world around us. But to avoid the tautology of organic things are living, living things are organic, I just wanted to flesh that out a little bit more. Organic molecules, like some of the ones that I've doodled here, are specifically molecules, chemical substances that are made up of carbon, hydrogen, and oxygen predominantly, though nitrogen, sulfur, phosphorus, and many other elements are often essential too, though in lower proportion. And it's because of their carbon hydrogen backbones that organic molecules can be a lot more complex, a lot more versatile than a lot of inorganic molecules. They can create interesting shapes that fit together and react with each other to create even more interesting and complex shapes. These structures that can react selectively with other structures. And that is something that's pretty essential to the building and the maintenance of life. But still, we're not quite at the point where we have a definition for life because, well, is it really enough to have a bunch of organic molecules floating in water on a rocky world? Is that a complete definition of life? And really, if that was the case, then we would have called it a day for the search of life for life in the universe when we first imaged and characterized Saturn's moon Titan. Now, Titan is absolutely fascinating, really cool, really interesting, don't get me wrong. And it's the only other world in our solar system that actually has liquid lakes and oceans at its surface, which makes it very special. And of course, it's extremely cold out there in the orbit of Saturn, so water is hard as rock, and instead the oceans are made up of organic molecules like methane and ethane. But Titan is a world that has tons of organic chemistry happening all the time, tons of these organic molecules floating about in a liquid solution. But for all of that, it really doesn't look much like life. It's still mostly just rocks and puddles. So that's sort of an unsatisfying definition. Life, as we understand it, has to be enclosed. It has to be separated from its environment somehow. It can't just be this diffuse soup floating around. That separation might take the form of some kind of membrane or wall. A membrane or wall that is semi-permeable, meaning that it can allow certain materials at certain times to pass across the membrane to exchange. And in doing so, it can allow the chemistry inside of the membrane to maintain some sort of stability or some sort of stable state, even as the environment around it is changing. This process, if you remember back to your high school biology, is what we call homeostasis, this ability to adapt to the environment while still keeping an identity or some kind of distinction between yourself and the outside. And yet, we're still not quite there with our definition. A bunch of organic molecules enclosed in a membrane or cell and maintaining some kind of homeostasis with its environment, we're getting close to a definition of life. But if this was all that we needed, then why wouldn't we consider a burlap sack full of dirt alive? There's still something missing for us. So it turns out that in order to be alive in the way that we recognize it, it really matters what's going on among the organic molecules inside that cell. It matters what processes are happening. So living things, as we understand them, they have what we call metabolism, wherein certain chemical reactions occur that will release energy that can then be used to facilitate chemical reactions that consume energy, and therefore wouldn't otherwise occur spontaneously. This coupling of energy producing to energy consuming reactions to the purpose of building and maintaining certain structures, this is really essential to our definition of life. But indeed, I think we have to go a step further. Life not only is maintaining its internal conditions and responding and adapting to its environment and performing metabolism, but it's also self-perpetuating. It's keeping itself going and it's replicating itself. It's making more of itself to put out into the world. This is something that's pretty essential to our definition of life and something that we're certainly very familiar with as so-called higher complex organisms, as well as among microorganisms. So with all of these conditions, we're now starting to approach a pretty solid definition of life, a definition that's a little bit more satisfying than, well, you'll know it when you see it, because life as we recognize it requires the complexity afforded by complex organic molecules. It requires separation or some distinction from the ambient environment. It has to have the ability to maintain its internal conditions and to adapt to external conditions. It has to perform metabolism, or the coupling of energy producing to energy consuming constructive chemical reactions. And it has to have this habit for self-perpetuation and making more of itself. But even with all of these conditions considered, our definition is still pretty imperfect. I'm sure if you think about it, there are probably tons of things that you can think of that you wouldn't necessarily call alive, and yet still satisfy most or all of the conditions that we've established. Viruses self-perpetuate, as do free RNA molecules, prions, wildfires. But there comes a point where you sort of have to throw up your hand and say, well, what can you do? This is perhaps as good a clear definition of life as we can get from a scientific perspective. A few years ago, in fact, I audited a class taught by a friend of mine who's now at the University of Washington. And he challenged the students in the class to come up with a definition for life, accumulating lists of conditions and caveats and exceptions that would help define life to our satisfaction as researchers. And the best thing that I could come up with was a little glib, perhaps in itself, but was this poem. Life is that which tries. Clouds and crystals may grow and evolve, stars may feed and die, a fire may spread its influence. But life is that which tries to survive, to grow, to learn, to adapt, to teach, and to be remembered in some way when it is gone. And I was satisfied with that for the most part, because at some point, what is life becomes this more philosophical, poetical question than a scientific question. But this is a science talk, not a poetry talk. So if this is what life is, a trying thing, a thing that maintains homeostasis, metabolizes, self-replicates, and all that, what does that mean for scientists who are looking for evidence of life in the universe? If you're an astronomer, looking for life generally means looking for a world that has evidence at its surface or in its atmosphere of processes that couldn't happen without life. On Earth, for example, there are certain molecules that you wouldn't see coexisting in the atmosphere, as they would react with each other and turn into something else. You wouldn't see them coexisting unless something was replenishing one or both of them rapidly, constantly, and at rates that outpace atmospheric circulation or geological weathering and processes such as this. Or perhaps there are even molecules that couldn't exist at all unless you had a living thing that performed metabolism, that could generate enough energy to put together a complex or inherently unstable structure. Now, starlight, or sunlight, depending on where you're standing, reaches the atmosphere and reaches those molecules in the atmosphere. And that sunlight represents a certain level of energy. It's carrying a certain amount of energy with it. When it reaches these molecules, that energy is imparted to the molecules. It interacts with their structure in such a way that some of it is used up and transformed into other forms of energy and re-emitted in different directions. And this is pretty critical for astronomers who are trying to study the atmospheres of planets, look at their composition and see if we can draw conclusions about the processes that produced those molecules. We have to be able to detect them. We have to be able to measure them in some way. So an astronomer far away from this hypothetical planet might be able to see the star that the world orbits. Maybe they use a telescope on the ground and have to spend a lot of time and effort accounting for the absorptions and emissions of their own planet's atmosphere. Maybe they cut to the chase and just use a space telescope of some description. But whatever method, they have some sort of instrument that allows them to collect the light from this star. They observe the light from that faraway star. And as it turns out, light from stars isn't really like the lights from a laser. Rather, it's light of a lot of different colors or energy levels or wavelengths, so that the astronomers can measure the intensity or the amount of light of all these different colors coming from the star. And this gives them a spectrum. It looks like it has kind of a shape to it. Some of those colors we might be able to see with our eyes, but some of them are too high or low energy, hence the need for some of these detectors that are hooked up to these telescopes. So astronomers can collect the spectrum of the star, and if the angle is right, and if the distance allows for it, and they can collect the star spectrum repeatedly over and over again over time, then they might be able to see how the spectrum changes. They might be able to see periodic changes because the star's light, as I mentioned, is interacting with the molecules in the atmosphere or at the surface of the planets that orbit it. So periodically, as these planets pass in front of the star, go behind the star, traverse just the edge of the star, you see the intensity, the brightness of light of certain colors going up or down at a rate according to that planet's orbital motion. And in some cases, the planet itself may even be bright enough, either emitting its own light of a certain color or reflecting some of the light from the star that it orbits. And these periodic changes to the spectrum of the star, to the intensity of these different colors of light, can be interpreted because we know that certain molecules interact with light of a certain energy level or certain color. So when we see the intensity of that color changing, we can say, aha, this planet has these specific molecules in its atmosphere. And of course, sometimes it's easier to do this when the star itself is smaller and dimmer, because then you can see the changes due to the planet's interactions with the planet's atmosphere a little bit more clearly. But we established earlier that life is pretty complicated and that there's more to the definition of life than just molecules floating around in some sort of ether. And astronomers can't be sure that the spectra that they are collecting and the way that those spectra are changing are actually showing them evidence of life unless they eliminate all of the other possible explanations for the presence or absence of certain molecules. And even if they are pretty sure that it's caused by life, that the molecules that they're seeing are the result of biological processes, then, well, then what? What else can you learn about that life? Because we don't just want to know that life is there. We want to know what it's like. We want to know what it looks like and what is it doing? What is the ecology like on the world that it occupies, and how is it shaping and changing the planet it lives on, or being changed by the planet it lives on? There's a lot of other questions that perhaps just remote sensing, observation at a great distance maybe isn't enough to answer. If we want to know the answers to these questions about what is life doing, how is it interacting with the world that hosts it, then we're going to need other tools. Geochemists, geologists, biologists, biochemists, and every sub-combination thereof, our approach is to study where those signature molecules in the atmosphere came from, how they were produced, and how different elements, different molecules, are cycled through planetary systems and different types of living things. So this diagram, this illustration, is one familiar to most biogeochemists. It's a simplified model of a rocky planet like Earth. You've got land, you've got bodies of water, you've got atmosphere, you've got weather systems, and I've done my best to cartoonily illustrate a temperature gradient coming up through the Earth's crust. You can imagine how an element, for example, say sulfur, might travel between these different parts of the planet. So some of the rocks in the crust have sulfur in them, which when heated by this temperature gradient, this heat source coming up from the Earth's interior, may change their form. They may be released from those rocks, from volcanoes and vents, and they may enter the atmosphere. And then that sulfur may interact with other elements while it's in the atmosphere. Maybe it will take on a different form, and it either may remain airborne, or it may rain out over land or over the oceans. It may re-enter the sediment, flow down a river toward the lakes and oceans, and on its way, participating in different kinds of chemical reactions, being incorporated into different structures, both living and non-living. And some of it ultimately may sink as sediment and accumulate and be buried deeper and deeper until it becomes incorporated into the rock once again, and the cycle continues. Every step of these cycles, every process, every transition determines the amount of sulfur and the form that it takes while it's in one reservoir or another. Geologists, chemists, biologists, bio-geo-chemists, we'll call them, are interested in the size and nature of each of these reservoirs, but they're also interested in the processes that occur within them and in transition between them. And because it's all connected, that you can expect changes in one part of the cycle to affect changes in other parts of the cycle, the composition and the properties of other reservoirs that are interconnected. And whether biology is involved or not, these processes are something that biogeochemists try to pull apart and try to understand in as much detail as they can. One of the major ways in which biogeochemists try to search for and characterize life is thus by measuring some of the properties, specifically the chemical and isotopic compositions of, and I'll talk about that in a moment, the different reservoirs and of the materials involved in the various processes and transitions between these reservoirs. So this is starting to get a little bit confusing. There's a lot of steps here. There's a lot of cycling. It seems like this very roundabout way of trying to probe, like, is there life and what is it doing? But you kind of have to, because all of these processes are interconnected and affecting one another. But to the purpose of actually finding answers to their questions, scientists often tend to simplify their picture of the world as much as possible, simplify their picture of all the world's biogeochemical cycles. And they divide the world into so many boxes, say an atmosphere box, an ocean box, a sedimentary pore fluids box, and illustrate them like so, as boxes with arrows moving between them. And those arrows represent the flux of a given material, typically a stable element like carbon or nitrogen or some such, between different boxes. For example, and I hope y'all can see my cursor, but I'll also say it out loud just in case. For example, these arrows pointing down from the atmosphere box might represent some amount of sulfur that is partially dissolved in rainwater as HS- or H2S. These are forms of hydrogen sulfide. Some amount of that sulfur enters the ocean. Some amount of it enters the sediments, either at the land or the ocean floor. And then there's some amount of exchange of sulfur, possibly as these forms of hydrogen sulfide, but also as sulfate, elemental sulfur, organically bound sulfur, oxidized sulfur species, all of these things moving between these different reservoirs. And some of this sulfur, ultimately, that isn't getting repeatedly cycled between these reservoirs might eventually become buried by successive generations of accumulating sediment, becoming incorporated into rocks that are deeper down in the crust. And perhaps eventually those rocks will be exhumed later or brought back to the surface when those rocks are thrust up as part of a mountain range, or perhaps they're buried deeper, and they get recycled through the mantle as the plate that they're on subducts. There's all these processes, but suffice to say, we can maybe simplify the world a little bit by representing these fluxes, representing these reservoirs as just boxes and arrows. And of course, the thing that we're interested in, of course, is the boxes that represent living organisms in the ocean and sediments and perhaps even in the atmosphere, but ran out of space in that box for this illustration. So continuing with this example of sulfur, because a certain amount of sulfur is going to be bound up in living organisms, and those organisms consume and excrete that sulfur in different forms according to their different metabolic strategies. At any given moment, there's going to be a certain amount of sulfur in each of these boxes, including the boxes for the living organisms. And regardless of the form that they're in, they're still sulfur, but there are different kinds of sulfur atoms that exist. It's not all just sulfur. There are different kinds of sulfur atoms that we call isotopes of sulfur, stable isotopes of sulfur. Now, isotopes, they're still sulfur. They've got the same number of protons. The individual atoms are almost the same, but they vary slightly in how heavy they are, how massive they are. And as it turns out, some of the processes, the transitions represented by these different arrows, some of these processes are mass dependent. That means that they care which isotopes of sulfur they use. And the reasons for that are myriad, but can sort of be simplified by the fact that, simplified is the concept that chemical bonds between heavier isotopes, stable isotopes at this point, tend to be more stable, sort of a lower energy state in the world than bonds between light isotopes. And for this reason, it's just subtly that much easier for example, cellular machinery to take up and break certain molecules apart. So, because of these mass-dependent, the mass-dependent nature of some of these processes, that means that the arrows that are passing upward in this diagram from the ocean and sediment reservoirs toward the atmosphere could represent processes that select for lighter isotopes of sulfur. The sulfur that is moving out of the ocean sediments and into the atmosphere is on balance lighter than what is left behind in the ocean and sediment reservoirs. Now, once you know how large these arrows are, as in how much sulfur is actually undergoing these various mass-dependent processes, and the size of these different reservoirs, you can try to figure out how heavy or how light each reservoir should be according if sulfur is moving, sulfur that's this light or this heavy is moving back and forth in this way. Or vice versa, and this is important to the search for life in the universe. If you can measure how heavy or how light different reservoirs are, or were in the past if you're looking at ancient rocks, then you can figure out perhaps what kinds of arrows, what kinds of processes were going on at the time that that rock formed. So, for example, if you see a difference in the heaviness or the lightness of the sulfur in the ocean reservoir and the atmosphere reservoir, the sediment reservoirs, then you can backtrack and figure out the extent to which, for example, bacterial sulfate reduction was operating in the world, or archaeal methane production, say if you're talking about carbon, or any number of other types of metabolic processes, these things that life is necessary to perform. So the heaviness or the lightness of the carbon, the oxygen, the sulfur, any of a dozen other elements in these different reservoirs is often expressed by scientists with this jaunty little lowercase delta symbol. Don't worry too much about how weird and squiggly it looks. And the selectiveness of these different processes for lighter or heavy isotopes is often represented by this this cheerful little epsilon, lowercase epsilon symbol. Now, this is a lot simpler than our picture of the overall world, but you can start to see how it would get kind of complicated, because we know that in reality, there are many more reservoirs than just atmosphere, ocean, sediment. And there are thousands and thousands of arrows moving between those reservoirs and cycling within those reservoirs. There's a bunch of different types of living organisms, different metabolic strategies, different types of chemistry that living things perform. And it can be difficult to tease it apart, not impossible, but through a combination of direct measurement in different types of environments, laboratory experiments, and numerical modeling, biogeochemists can use elemental molecular composition and isotope composition, along with knowledge of how biological or living processes differ from abiological or non-living processes. They can use all of this to figure out whether or not life was happening in a particular time and place, and also what that life was doing, what substances it was living on, and how it was responding to its environment. And this isn't foolproof. Just like astronomers have to rule out all of the possible non-living explanations for the presence of methane or ozone or things like that in the atmosphere of a planet that they believe could harbor life, theoretically, a biogeochemist also has to separate out all of the non-living and living and sort of kind of living explanations that might exist for the properties of these different reservoirs. So it's messy. It's not always definitive, and it's hard to take a measurement of, say, the sulfur isotope composition of a piece of pyrite in a billion-year-old rock and be sure that the measurement that you've made, that the lightness or the heaviness of the sulfur in that pyrite is the result of that sulfur being derived from what used to be a sulfate-reducing bacterium. And to be sure that that sulfur was metabolized in a certain way, processed in a certain way by this organism that you know nothing else about. And this is, of course, true for many other elements. You can swap out sulfur with nitrogen, with strontium, and all of these other things. It's difficult to draw these conclusions and rule out non-living processes, and to be sure of any conclusions that you might draw about the ecology of the world where that pyrite formed. So really, if you really, really want to be sure that what you're looking at, either in deep time on the Earth or at the surface of another planet, is truly evidence of a particular kind of life at a particular time and place, then what you really want is a good fossil. And I personally love fossils. What person doesn't? Fossils are cool. And they're great for telling us about past life, but they too can be difficult to find and difficult to interpret, especially because of how hard it is to become a fossil, particularly if you're a microorganism, like, for example, a little sulfate-reducing bacterium. In order to become a fossil, this little sulfate-reducing bacterium has to make it down to the seafloor without being consumed by any number of other voracious microbes and other life forms. It has to be buried relatively rapidly. It has to sort of maintain some of its structural integrity, even as fluids, either rainwater or groundwater or seawater, percolate through those sediments. It has to survive the lithification, the formation of rock. It has to survive being recrystallized. And perhaps crystals grow on or around the template of the organism itself, if you're really lucky. And then that structure has to survive long enough to become a chunk of rock that is exposed at the surface of a planet that a researcher can find and look at. And it's difficult to try to back out the ecological history of the planet where you found this rock from something that often looks like a little schmutz, the surface or of a rock. Of course, it doesn't always have to be the individual fossil itself. It can be more of a trace fossil, if you will. Microorganisms often form accumulations at the bottoms of seafloors, lakes, dirty bathtubs, and they collect and they stick to infalling bits of sediment, and then another layer comes and more microbes show up, and it gets buried deeper and deeper, and this happens over time until eventually you have a rock with a certain distinctive pattern in it. And this pattern would be difficult to achieve if you didn't have that mat of microbes there. So perhaps as a geologist, you see that in the field and you think, man, like that really looks like something a microbe would make or build up. So you take a piece of that rock and you subject it to all these different analyses. You drill a little bit of it out, make a powder that you can then process and use to measure the isotopic and chemical compositions of the rock. You put it under a hand lens, then you put it under increasingly powerful microscopes to look at the microscopic details and see if you can find other physical or chemical evidence for microbial activity preserved in this rock. You might shoot lasers or other more broad spectrum light sources at it, and just like the astronomers are interested in how the molecules in another planet's atmosphere might interact with different colors of light, geologists are indeed interested, geochemists, don't want to be exclusive, are indeed interested in how the chemistry of a rock interacts with different colors of light. And I won't go into too much detail here, and we can talk about exactly what kinds of light and what instrumentation and things are used for this purpose. But one of the things that I do pretty frequently in my own work is I take these rocks that have some sort of more macro or mesoscale evidence of some kind of structure that would be really hard to achieve without microorganisms. And I try to create maps of their chemical composition, sometimes their isotopic composition. And I turn these spectra from a grid of points across the surface of these samples into a map of certain spectral features and their intensity, their strength, in a 2D representation. This is pretty powerful. This is pretty awesome to be able to make a map of these things, because then you can look for differences in the composition between what you suspect to be a biogenic structure, a structure created by a living thing, and an abiogenic structure created by abiological processes, just normal sedimentation at the seafloor without the activity of cells. In fact, this is a photograph. It's about 2 centimeters across or so from left to right of your screen. This is an image of one of these sort of suspected microbial accumulations in a rock that's actually between 1.9 and 2 billion years old, from a rock formation relatively close to Thunder Bay, Ontario. So unfortunately, the nice round blobby looking things you see in this image are not fossils. They're not individual organisms. In fact, those are something we call ooids, which we can talk about in more detail later if people are interested. But the actual likely biogenic structures that we're seeing here are these sort of domes and columns, these sort of fingers that are stretching from the bottom left to the upper right of your screen. These shapes, as it turns out, are pretty hard to make without the assistance of some kind of microbial mat or biofilm trapping and binding and accumulating sediment, especially near these margins where you see that you've got layers of sediment that have been stuck there well past the angle of repose, well past the angle where they should just roll off according to gravity. Something is sticking them there. And the thing that is sticking them there is sticking them there in a very sort of uneven, blobby, pinching and bulging kind of growth habit. Biology loves pinching and bulging and being blobby and being somewhat irregular in a way that just normal crystal growth, abiogenic crystal growth isn't really. And indeed, when you zoom in this same sample, the scale bar in the middle of the screen says 100 mu m. That means 100 micrometers, or 100 millionths of a meter. That's the length of that line right there. And you can see sort of on the order of 100 micrometers, 200 micrometers in length, and maybe only a few scant 10 or less micrometers across, all these little hair-like things, these little filaments. These filaments are microbial fossils. Indeed, we suspect that they are 2 billion year old form of cyanobacteria, which is really interesting, especially considering the important role that cyanobacteria play even now in the modern world in generating oxygen, gaseous oxygen that fills the ocean and changes the composition of the atmosphere. And one of the reasons, additionally, that we're pretty convinced that this is cyanobacteria and not something else is because we're able to couple these images with compositional information and contextual information about the part of the world and the nature and the history of the rocks where these little microorganisms were found. Now, mostly thus far, I've been talking about single-celled life, bacteria, archaea, these tiny little germ like things. And you might ask, Cecilia, typically when we hear someone talk about paleontology and ancient life, they're talking about stuff like this, these awesome giant skeletons, these huge complex creatures. And certainly they're very impressive. I love macrofossils. I love dinosaurs. I love bones. I love shells. They're fascinating. But usually when we talk about the search for life in the universe, we tend not to be talking about these big kind of charismatic, bony, shelly organisms. And the main reason for this is that the bigger and more complicated your life form is, the more it needs from its environment, the more conditions have to be satisfied in order for it to exist. Because indeed, all of these, in order to survive on Mars, all of these big, meaty dinosaurs would have had space helmets of some sort so that they could have some source of oxygen that they could breathe, because these big organisms take a lot of energy to build and to maintain themselves. And we went back at the beginning, I talked about the importance of metabolism, the ability to couple very energy generating reactions with energy consumptive reactions. And as it turns out, some of the most effective, some of the most bang for your buck energy producing reactions involve oxygen. So if you're on a world without oxygen, then it's pretty hard to get dinosaurs. And you might ask, well, Cecilia, the space is very big. There's a lot of planets and worlds. There's got to be some worlds out there with dinosaurs with space helmets. Why don't we look for those? Why aren't we finding those? And why are you so interested in these tiny little wiggly bits? And well, I won't go into a ton of detail about this, but I like to say that Earth is the only place in the known universe where there's life. So we sort of have to take our cues and look at the example that Earth gives us. And in the whole timeline of Earth history, the Earth is about 4.5 billion years old. That's what 4.5 GA means here. That's just geologist shorthand for a billion years ago. In that whole timeline of Earth history, really the microorganisms tend to win out. from the time that Earth formed, sort of the last, the end of accretion, the moon forming impact, through the formation of, or at least to our knowledge, the earliest appearance of liquid water at the surface, to the appearance of the first sort of lighter reservoir of carbon in ancient rocks, all the way through, let me just skip ahead here, all the way through about 635 to 541 million years ago. We have no evidence that there were bony, shelly, big, complex organisms. All of these little tabs along the timeline actually represent evidence of, or, well, varying levels of convincingness, evidence for microorganisms. And you can see that they're still placed pretty few and far between. I talked about how difficult it is to create a microfossil, how difficult it is to tease apart the geochemical signature of a biological process versus an abiological process. And of course, the older the rocks get, the more stuff has happened to them, the more difficult it is to read that geologic record for the planet. So in some ways, ancient Earth is a pretty good, or searching for life in the geological records of ancient Earth is a pretty suitable analog for searching for life on other rocky planets. And of course, it's not until this 635 to 541 million year sort of age range, going into the 541 to 485 million years ago age range, that you really start to get complex, bony, shelly organisms. It's not till 470 million years ago that we start to get land plants, eventually vascular plants, bony fishes, conifers, flying dinosaurs, deciduous trees, terror birds, giant sloths, humans. They don't show up till way, way late. So I think that it's probably more likely that when we do find evidence of life in the universe, I think that it's probably more likely to be this simple, humble microbial life, which dominated most of the history of life on our own planet, and are virtually ubiquitous in every possible set of environmental conditions that exist at the surface and in the subsurface of the planet. Much more likely to be those than perhaps this cluster of big, charismatic, interesting fauna over here, sort of in the last 10, 11% of Earth history. So if there's no life for the first 10% of the timeline, and multicellular life in the last 10 to 11% or so, then that's almost 80% of Earth history. That's microbes world. The microbes are in charge. So when we're searching for life on another world, we've moved beyond the search for life with remote sensing techniques, telescopes, the interpretation of spectra of distant stars and planets. When we've actually gotten to the point where we can get to the surface and we can look at the rocks, we can look at the dirt, we can sniff the air, sample whatever fluids there might be at the surface, then I think that we'll have to turn to these biogeochemical techniques, the things that we use to study extinct and extant life on our own planet, in order to understand, in order to define, describe, and really fully appreciate life on other worlds. This image is one of my favorite images that I took early on in my PhD when I was first learning how to use a fluorescence microscope, where I washed some rock samples that I'd been working with that I'd been incubating in the lab with water from a deep, kilometer deep well. I washed it with a fluorescent dye, a type of dye where the fluorophores, the fluorescent molecules, stick to parts of the DNA, the genetic information contained in each cell. So every glowing pinpoint you see here is a living thing, or a previously living thing before I dunked it in the solution necessary to mount it on the microscope slide. And I just like this image so much because I really think it looks rather like kind of an abstract starry night sky. And there's something, sorry to go back to the poetry, but there's something rather poetic about this visual comparison between the living and the non-living, the similarity between the two and the interconnectedness of them that I just find very inspiring. So one day when we're on this other planet, we're exploring the surface, the tools that we bring with us, the toolbox, the things that we use to assess, is it life, is it not? They're going to look a lot like the toolbox we use to assess what life was like on ancient Earth. And we'll pair that with all of the context that we collect from the astronomical, astrophysical data, from the remote orbital surveys that we'll inevitably do. But I hope eventually that we'll really be able to go down there, pick up a handful of dirt, and look at it as closely and as in-depth as possible. And maybe we'll bring along some kind of small mass spectrometer as well to get its isotopic composition, because unless we combine all of these different tools, we draw from all of these different data sets, it's going to be really hard to find something that's definitively life. With that, I know that I'm over time and perhaps have been a bit overindulgent with the poetry, the waxing poetic. So I'll go ahead and finish up there. My name is Cecilia Sanders, PhD candidate in geobiology. I'm interested in all questions of geobiology, planetary evolution, and in general, anything that's wiggly and lives on or is incorporated inside of a rock. If you're interested in contacting me directly to ask further questions about my work or anything tangential to it, my email is csanders@caltech.edu. And with that, I believe we can open things up for the Q&A.
Great, thank you, Cecilia. That was great. My green screen is showing here.
Shall I go ahead and end the slides?
Yeah, if you would. And we can welcome the rest of our team. Everybody go ahead and bring your cameras back on. Awesome, thank you. We had a number of great questions. I urge our audience members to continue to ask questions in the chat. We're monitoring it, and we can address them even if we don't get to yours immediately. We'll try and get to it over the course of the next hour. Before we go any farther, first of all, that was great. Thank you, Cecilia, for joining us. It was really awesome. Very different from a lot of our past talks in terms of subject matter, but extremely interesting. So thank you. So before we start getting to the questions, just so everyone knows who everyone on our panel is, as I mentioned, these are all members of the Caltech Astronomy or the Caltech Geology and Planetary Sciences Department who are representatives here. And I'll give each one of you a minute or so to introduce yourself and the kind of science that you do. I already said I'm Dr. Cameron Hummels. I do research in computer simulations of how galaxies form and evolve and the regions surrounding galaxies from just after the Big Bang to present, and try and better understand that through theoretical modeling. Who's Dylan? I invite you next.
Hi everybody, my name is Dylan, and I am a fourth year, soon to be fifth year, PhD candidate in astronomy at Caltech. And I study mainly radio astronomy. So I use large radio telescopes and arrays of radio telescopes, such as the Very Large Array, to study stars as they're forming and also stars as they're blowing up. And I also work in a little bit in black holes as well, as they're eating the gas that spirals in, and sometimes eating other stars that are spiraling in. For fun, lately during the pandemic, I've been hanging out with my pet bunnies a lot. So I've just been flopping on the floor right next to them. And I've trained Eloise at least to start giving me nose boops in the last week, which I'm very, very proud of. But aside from that, I've also recently been rediscovering my love of bushwhacking and also just scrambling in our local mountains, trying to get some hiking in while not infecting other people with coronavirus and hopefully not getting infected myself. And yeah, besides that, I've been doing a little bit of work with grad student governments. Like I'm on the Physics, Math, and Astronomy Student Advisory Board and the Graduate Student Council, as well as this group called Caltech for Affordable Healthcare. And we've been working on things like trying to get expanded healthcare access for grad students during the pandemic, as well as advocating for the rights of our Black grad students.
Great.
Which is
Great. Teaching your bunnies to boop noses with you. I like that. Nicole, would you like to introduce yourself?
Yeah, hi, I'm Nicole Wallach. I am a PhD candidate in planetary science. I'm also a fourth year going into being a fifth year. I don't really want to think about that too hard, but I guess that's what it is. So mostly what I work on is the atmospheres of exoplanets. So I use ground and space-based telescopes, like Cecilia had mentioned in her talk, to study the atmospheres of exoplanets and also of a class of stars called ultra-cool brown dwarfs, which feel free to ask me more about. It's actually the background, my Zoom background, putting some science there. So I can talk more about what these ultra-cool brown dwarfs are. We're working on that. I also do some direct imaging work to take real pictures of protoplanetary disks. So the disks that form around stars, the material that planets form out of. So just trying to understand a lot more about how planets form and evolve. I guess since we're doing the for fun part, I also studied film in undergrad. So I've been using the pandemic to kind of catch up on some cinema stuff and trying to watch a lot more sci-fi and trying to turn off the part of my brain that's science focused so I can actually enjoy that sci-fi sometimes. Some movies are better than others. And if you have any questions about that also, I will rant about that.
Yeah, that's great. When I think of bad science movies, I think of The Core.
Yes.
I think that's up there for being one of the worst science sci-fi movies.
The geophysics department does that actually. So the geophysics department so Cecilia and I are actually in the same department, geological and planetary sciences, and the geophysicists will watch The Core and just get really mad about it, basically, is mostly what they do. Like every year. You can tell that they're doing it because they're yelling in the hallways. It's kind of a mess.
That's great. Andreas, would you like to introduce yourself?
Sure. My name is Andreas Feist. I was born in Zurich, Switzerland, and I came to Caltech in 2015. And now I'm an assistant scientist at Caltech. My research focuses mostly on the very, very first galaxies that were formed in our universe. And I try to understand how they formed, how they evolved, and how they grew over time. And also what's really cool, how they move, how they rotate, and how they look like. And as you can see on the picture in my background, they don't look like the Milky Way. I'm the lead of a large collaboration that uses the ALMA radio telescope in Chile to look at those galaxies. Specifically, we look at the dust. So how dusty are those galaxies? Because that's very interesting because dust is built up over time. So when we know how much dust the galaxies have, we know how mature those galaxies are and how evolved. For the fun part, I'm doing also a lot of outreach, which is fun in and around Los Angeles and also in Switzerland. I was playing piano concerts, at the moment not that many anymore. And if you have questions, then that's mostly about Swiss chocolate and, as I said, galaxy evolution.
Swiss chocolate. I have a lot of questions about Swiss chocolate for you later.
We can talk offline about that.
Okay, so we've had a number of great questions, both in the YouTube channel as well as beforehand. So I will dive into some of those. Specifically, let's take a few questions that we heard about the talk. So there were a couple of questions we received about the possibility of non-carbon based life, like silicon based life, or other things. Can you speak to that at all, Cecilia?
A little bit, yeah. This idea of silicon-based life instead of carbon-based life is pretty old and pretty popular. Something that I believe is addressed multiple times in Star Trek. And I believe the reason for this is because silicon is it's certainly not the same element as carbon, but it's capable of forming similar arrangements of bonds. It's capable of forming these complex, interesting structures. It forms, crystals. And so for this reason, I believe it's captured a lot of people's imagination as a possible alternative to carbon in the construction of complex and sort of reactivity selective molecules that are necessary to build the structures of life. The trouble is that, to our knowledge, as scientists who do experiments in the lab as well as observe biogeochemical processes out in the world, silicon isn't really capable of making structures analogous to the semi-permeable membranes, the membrane transport proteins, a lot of these things that are pretty essential to our understanding of what a cell is and what life is. Silicon can form complex structures, but those structures tend to end up looking like aluminosilicate minerals. They look like rocks, as opposed to things that can perform metabolism and maintain homeostasis and stuff. And so it's a little bit difficult to imagine a world populated by silicon-based life forms. We probably wouldn't recognize it as life, as I attempted to describe it, and many other scientists besides myself have attempted to describe it. That said, silicon participates in a lot of really interesting chemical processes, even though they're not living. Minerals are really fascinating structures, and are often in their own way essential to certain life forms, as growth substrates and as suppliers of trace metals and things like that. So yeah, I think that long answer to a short question, silicon-based life, probably not a thing unless we expand our definition of what life is. But it's quite possible that there are other, that you can create structures analogous to the structures of a cell, the cellular machinery for metabolism, using molecules other than the basic biomolecules, carbohydrates, nucleic acids, proteins, things like that. There are other types of molecules, most of them, again, organic, carbon, hydrogen, oxygen based, that can produce some of these structures. And indeed, there's a lot of work being done at a lot of institutions to simulate those processes.
Excellent. Thank you. Some related questions. Again, for the related to the topic, would a significant quantity of oxygen on a suitably located exoplanet not be a sufficient indicator of life on that planet? So is like are high concentrations of oxygen a biosignature that we could rely on, or is that not something that we could rely on as detecting life?
Yeah, I could address this in part, and then I think that Nicole, who studies planetary atmospheres, should add to this answer. But I would say no, as the short as the short version. But the reason that oxygen is so interesting to scientists, to researchers who are looking for life in the universe, is because on Earth, our oxygen-rich atmosphere and our mostly oxygenated oceans they derive their oxygen from photosynthetic life. So I glazed over it really quickly in my presentation when I was doing the step through of the history of life on Earth. But about at the midpoint history at around 2.5 billion years ago, we start seeing evidence in the geochemical record that Earth's oceans and atmosphere started accumulating oxygen. You start seeing you stop seeing a large abundance of certain minerals and certain metals in a more reduced form. Things are oxidized. You've got rust. Basically, the world gets rusty, and this coincides with what we believe from the geobiological record to be the rise of microorganisms capable of oxygenic photosynthesis. We put two and two together. It seems that around 2.5 billion years ago, finally, oxygenic photosynthesizers were able to fill up every other possible sink for oxygen that the world had, to the point that it just starts accumulating in the atmosphere. So if you see a lot of oxygen in another planet's atmosphere, it would certainly be tempting to conclude that, like the Earth, this oxygen is the result of photosynthesis. It's the result of plants or algae or cyanobacteria or something like that living at the surface, and perhaps passing that same milestone that the Earth did about 2.5 billion years ago. But there are other processes, that inorganic, or rather abiotic, non-living processes that can also generate oxygen. And my understanding is a lot of this has to do with the interaction of the star's light with perhaps water at the planet's surface or in the planet's atmosphere. It can split water, and once you have free oxygen bopping around in the atmosphere, it's difficult at a distance to like distinguish that oxygen from the oxygen generated by photosynthetic microorganisms, unless you can also collect information about the isotopic composition of that oxygen. So with that, I think, Nicole, you're nodding. I think you should weigh in.
Yeah, so I'm an observational astronomer, I should say. So a lot of when I get these kinds of questions or hear these kinds of questions, I think, how feasible is it to actually be able to make these measurements? And the short answer is not very for oxygen. So different molecules, because of the way they interact with light, become easier or harder to detect in atmospheres. So oxygen, O2, is actually incredibly difficult to detect just in general. And the only thing that we think, there's actually a paper recently out a few months ago that was talking about the ability to detect O2 using the James Webb Space Telescope. So the James Webb Space Telescope is a telescope 6.5 meter long diameter, I should say, primary mirror. It's a massive telescope that's going to go into space next year, hopefully, assuming there's no more delays. But it's a massive undertaking. And whenever you see a paper that says JWST will solve all our problems, you know it's been very hard in the past to get these measurements, and we actually don't have any. And it just has to do with the way that O2 is, the molecule that is O2. So even setting aside if the value was very high for your O2 concentration, how would we actually be able to detect it? And we can kind of do it possibly according to this one paper for one sort of TRAPPIST-like system, possibly, that's close to the Earth. So it doesn't seem like it's something that we can really get. These biosignatures, the signatures of biological life, are very hard to detect, just because of the nature of the molecules, most of them, the ones that you could, if you could disentangle them from non-biological sources. So I think the long and the short answer is, unfortunately, even if we could detect it, we're not sure, and we can't detect it.
This was actually the subject of my junior thesis as an undergraduate, was not actually attempting to detect oxygen in the atmospheres of Earth-like, so rocky, Earth-size planets around red dwarfs, or M dwarfs rather, but actually just simulating that data. So taking model spectra of these smaller, dimmer stars and simulating the interaction of that star's light with the annulus of the atmosphere around the disk of the planet as it passed in front of the star, or passed between the theoretical telescope and the star itself. And then adding to that simulated noise, just the general noise that you get from the instrumentation, but also noise as the result of overprint from our own atmosphere if we're using an Earth-based telescope, and fluctuations in temperature, all of these things. And my whole project, the whole attempt, was to see if we could tease out the signal of a super oxygen-rich atmosphere of a rocky planet around this distant star. Just hypothetically, not working with real data, just if we idealized all of the other possible variables and conditions. And the answer that I came up with, again, as a junior, so take it with a grain of salt, was, man, this is really hard, so probably not, unless I tweaked the parameters to give us an absurdly large telescope, and place that telescope beyond the influence of Earth's oxygen and water-rich atmosphere. So yeah, it's a really interesting problem, both of detection, but also of like interpretation. How convincing is an oxygen detection?
Great, thanks guys. Another question that's related to life, but on an exoplanet. Could life exist on an exoplanet orbiting an O-type star? Dylan or Nicole, would you guys be willing to?
Sure. Yeah, so just for some background, an O-type star is an extremely massive star. So I don't remember exactly what the line is between B-type stars and O-type stars, but it's something like 15 times the mass of the sun, or 20 times the mass of the sun, or something like that. And so what happens when you have a star that massive is it runs out of fuel extremely quickly. And remember that Cecilia was talking about in her lecture that there wasn't really life on Earth for the first 10% of Earth's life. Right? So that's like hundreds of millions of years. And an O-type star, by contrast, lives about 1 million years, or like 2 million years. So it's likely that life won't have enough time to evolve before, if it's anything like life on Earth, before the O-type star blows up. So the answer is probably not.
Okay. Oh, now I'm a disembodied head. All right. Well, that's okay for now. I'll fix things here. We'll take a question that's totally unrelated to the talk topic for tonight, but was just asked. For Andreas, how do earlier galaxies differ from later ones?
That's a very interesting question. So basically, galaxies, they grow like we do, right? So when they are formed, they're very small. They don't have a lot of stellar mass, so they don't have a lot of stars. And then they grow over time. And the question is, how do they grow? And they can grow, for example, via gas that's funneled onto the galaxy. And with this gas, stars are formed. So you have basically a lot of Orion nebula in the galaxies that are forming stars at a very, very high rate, about 100 times to 1,000 times more than our Milky Way. Or you can collide galaxies, and then they're basically disturbed and they grow, right? And so depending on how they do that, they look very differently. And so in the early universe, you have a very high density of galaxies because the universe was smaller and then expands over time. So you have a lot of collisions. So those galaxies, they look, they don't really have a defined shape. So they are colliding, they are very irregular, a little bit like the galaxy here in the background. And then over time, they start to form a disk, and they don't have collisions anymore. So they can stabilize their disk, and they're becoming those nice Milky Way-like disk galaxies that we see today. And this whole process takes about 12 billion years. So the first galaxies are formed maybe 500 million years after the Big Bang. They're very irregular. And then maybe 3 to 4 billion years later, they are forming their disk structure. But it can still happen that they, at some point, collide with another galaxy. And then instead of evolving into a disk structure, they evolve in what we call an elliptical galaxy, which is basically just a sphere of stars. And those galaxies, they usually don't form any stars anymore. So we call them red and dead because the stars are old, and so their light is red and not blue anymore. And they form these elliptical galaxies.
Awesome. Thank you, Andreas. That was pretty comprehensive. Excellent. All right, so looks like there's fireworks outside. I guess people are still celebrating the 4th. So a few questions. There's reference in the chat to signals being received in China every 16 days, radio pulses being received from another galaxy. Dylan, you're the resident radio expert. Can you take on this question?
Yeah, so what this is referring to, first of all, the telescope in China is called FAST, or the 500-meter aperture spherical telescope. I think that's what it stands for. And what FAST is really good at is it's extremely sensitive. Like it's a huge dish that's basically built into like a mountain valley. And what it does is it stares at one part of the sky and it collects like a huge amount of light that's coming from that direction of the sky. And so it can see things that are really faint. And recently what FAST has done is it's found like thousands of bursts from this one object called a repeating fast radio burst. Okay. And there have been some results that didn't actually originally come out of FAST. It came out of a Canadian experiment called CHIME that showed that one of these fast radio bursts, one of these repeaters, has periodicity. So what that means in this case is there's times when the fast radio burst repeater is on and there's times when it's off. And so when it's on, you see lots and lots of bursts in that window, but when it's off, you see no bursts. And so they were able to statistically show that this effect is real, and it has something to do with whatever is producing these bursts. Now, separately, there's been lots of speculation as to what fast radio bursts actually are. There have been like hundreds of theory papers that have been written proposing like different mechanisms. And these have ranged from bursts from neutron stars, from these highly magnetized neutron stars called magnetars, to even like aliens, right? And aliens, of course, are the like really attention-grabbing headlines. And recently, there's been some evidence that it's likely actually this highly magnetized neutron star that's the cause of these fast radio bursts. There's actually one particular burst, or actually two, that happened in very quick succession that happened in our galaxy that was recently localized by both the Canadian instrument CHIME as well as STARE, which is an instrument that was built by a grad student at Caltech. And this instrument found, or these two instruments found that this burst happened in a location of the sky at which contains one of these highly magnetized neutron stars at the exact same time that that neutron star happened to be flaring in the gamma rays. And so it's likely that both the fast radio burst and the gamma ray flare are related, just because you would have to be extremely, extremely lucky, and you have to be a huge coincidence for those two to happen in the same direction of the sky at the same time. And so, you know, if this fast radio burst is the same as the fast radio bursts that have been seen from repeaters, and if the localization is correct, which it probably is, that part, then it's likely that these signals that FAST is seeing from other galaxies are from these neutron stars that are highly magnetized. At least that's the state of the field right now.
Excellent. Thank you. That was excellent. All right. I like this question. It's a group question. What are the panel's favorite solutions to the Fermi paradox? And for those of you who aren't aware, the Fermi paradox was postulated in the last 70 or so years by Enrico Fermi, a famous physicist. And that was, you know, if you assume even a small chance of life developing or intelligent life developing, there are, you know, billions of stars and probably billions of planets in our galaxy alone. Why haven't we, you know, the main question is, where is everybody? Where are all the other sources of life? If you know, even if life exists in a very small fraction of planetary systems, like one billionth of a system, the universe should be teeming with life. So why haven't we yet discovered it? So what are the rest of the panels, the panelists' opinions on why we haven't?
Do you want to start, Nicole, or shall I?
Oh, either way.
Sure. So I'll go ahead and weigh in on it from this sort of history of life, geobiological perspective. So I think that one of the central problems with the Fermi paradox is this assumption that there's some kind of natural progression for life from single-celled organism to spacefaring civilization. And it's just not. So I know that part of the argument with the Fermi paradox is that, oh, because the universe is such a big place, everything's got to happen somewhere. Like, of course, there's going to be intelligent life somewhere else. But like this, I think that I don't know if I'm particularly convinced of that. No one, like, an intelligent civilization doesn't have to become a spacefaring civilization. It doesn't have to become either a conquering civilization or a civilization concerned with being emissaries. That's, you know, it's perfectly possible to develop complex life that has no interest in doing the things that we as humans do. And so I think that just as I wouldn't necessarily expect to find an elephant eating banana leaves on another somewhere else in the universe, just because the universe is a big place, I wouldn't necessarily expect to find expect that there should be another kind of intelligent life in the universe that also wants to reach out and talk. Additionally, I think that a big problem with becoming a spacefaring civilization is just that space is big. It's so big. It's so just unfathomably, like, ridiculously big. It's really hard to get from point A to point B. And there's some theoretical ideas, just ideas, not anything that's executable, for how to travel faster than the speed of light or go through wormholes and things like that. But we don't really have any reason to believe that that's possible. And so if you're just moving through space at as fast as the technology that we can conceive of allow you to go, it's just so, it would take so improbably long to be able to reach another world. Definitely the organisms that get on the ship are not going to be the same organisms that get off the ship at the other end. And of course, perhaps there's such a thing as infinite beings, but again, back to the problem of, if you're an infinite being, why would we assume that your intrinsic desire is to just go on interminably long car rides? So I think that Fermi's paradox, the question that it poses, is perhaps a wrong question. Not why haven't I'm less concerned about why haven't we been visited by intelligent life, and more interested in why is there intelligent life here at all? What's special about this time, this place in the universe? What combination of very specific and very important events had to happen in this right sequence in order to result in the way that we are today? I think that's perhaps a tackleable question, and will perhaps help us understand how common, how possible things like us are. But yeah, I think one of my I'm a huge fan of science fiction and fantasy, and one of my favorite things is imagining how different civilizations on other worlds might be. They're almost guaranteed not to want the same things as we want or think the way that we think, which brings us back to poetry and philosophy rather than science. But yeah, that would be my answer.
That's certainly a well-reasoned perspective on all of this. I would also add, though, that we could be in contact with other civilizations or other intelligent life without having them have to travel all the way to us to get here by signals, radio signals, as was being discussed, potentially from fast radio bursts and so on and so forth. But even the speed of light isn't that fast either. To traverse the width of the galaxy, which is only one small speck in the larger universe, takes, to our eyes, an interminably long period of time to traverse that, even at the speed of light. So, Nicole, you wanted to add on to that? I mean, we can hear from everyone on this topic. I'm sure everyone has opinions about this, but.
Pretty much the same as your guys is mostly. It takes time for even radio signals to get places. So our sphere of influence, if you want to call it, of the Earth is not that big in the grand scheme of the universe. So the same reason that we're not seeing other civilizations could be the same reason that they're not seeing us. Everything is so far away and takes so long to get there. If we're talking about actual physical things that have gone off of the Earth, we're not talking about very far. We're talking barely outside our own solar system. So physical things take too long. Even radio signals, television signals, things like that, that could show, yep, that's absolutely intelligent life. There's no way that I Love Lucy came from, a not biological thing. It still takes time to get to another place to be able to be heard. So I don't really think it's I think, like Cecilia said, it's not exactly asking, I think, an interesting question necessarily. I think it's very obvious for the most part, kind of why, but doesn't stop us from kind of thinking about what those civilizations are like. And I think that's the kind of cool part. We have absolutely no idea, so just be poetic about it. Why not?
And the universe is also only so old.
Yeah.
Just thinking about that as you were talking. It took 4.5 billion years for us to get where we are. The universe is what, 13 billion years old? So, how long does it take to create I Love Lucy? Maybe 4.5 billion years is the average. I don't know.
Worth it though, right? Worth the wait, I guess.
Well, but I think what some critics of that would state is simply like the rate at which our technology has been increasing in recent times. The difference between the world in which we live today versus the world of 1,000 years ago is dramatic in terms of our ability to perceive the surrounding universe. Our understanding of the universe has improved by leaps and bounds beyond what they thought in the Dark Ages. And 1,000 years is nothing compared to the whole buildup of the biotic life on Earth or the solar system as a whole. So who knows at what level our technology would be in another 1,000 years or 10,000 years. I mean, I can't even figure out what the next OS X from Mac is going to look like in a year. So let alone what we're going to be able to do and go to space in 5 years or 10 years.
Yeah, you can also turn this around, right? And say, how long are we, when was the first time we were able to actually receive radio waves, right? And that was like, I don't know, 70 years. So put this into perspective of like, I don't know, 1 million year. And then it's basically, we just turned on the TV, right? So maybe we just don't see.
Dylan?
Yeah, just to echo that, let's say that there was some sort of intelligent civilization that wanted to contact us. To say that that was the case, they would have had to, unless they were extremely, extremely close to Earth, they would have had to decide, like, oh, look, the civilization just built the pyramids. Let's blast radio waves at them and hope that, like, in a few thousand years, they'll invent radio receivers that are able to, receive the things that we're blasting. And so, our sphere of influence, like I think Nicole was saying, really is quite small. Like 70 light years. There are only, I don't know, probably a few hundred or a few thousand stars within there, right? And if you think that intelligent life evolves, if that's a 1 in a million kind of chance or a 1 in a billion kind of chance, like a few hundred or a few thousand is not going to likely get you another intelligent civilization.
So in general, the feeling that I'm getting from all of you is the very optimistic feeling that we haven't been around long enough, we haven't had the capability to look long enough, as opposed to the counter side to that, which is what some scientists hold, which is that we haven't discovered life because technologically capable civilizations will self-destruct in a short period of time. And so those would have got, I'm just saying this for the purposes of full disclosure to our audience of what some of the theories that people hold.
Actually, that you mentioned that, actually, because there are some papers that have been written about what a world that destructed itself due to some nuclear holocaust, for example, would look like, and whether we could detect that or not.
Oh, cool.
Yeah, they're astrobiological studies mostly, and they're really interesting and super cool and completely just hypothetical. And even those that are like, all right, there's some like global catastrophe that's very obviously formed by a civilization, what would that look like? And even that, the answer is we would not be able to see it for very long, even if we could. So
Hmm.
I know that's not optimistic, but there are studies that have been done on it. So I think someone's thought about this in a great deal.
You know how depressing that is when you finally find a civilization, but you know, actually, it's gone.
Yeah, it's like Wall-E. Poor Wall-E.
This is the thing. It's like, that's like, it's certainly like a very cynical view, like, oh, they must destroy themselves, but it's also this very kind of, I don't know, like weirdly like myopic imperialist view of human civilization. Like on our own planet, you know, we're not culturally a monolith. And while there are some civilizations that like ended up being incredibly warlike and either and expanding until they were too big to like maintain themselves and such, there's also plenty of civilizations that didn't do that, and some of that interestingly is like a direct result of where in the world they ended up settling and what the availability of certain essential natural resources was. Like it all, you know, it goes back to the geology. But it's I think that again, like the assumption that in order to be this like technologically advanced spacefaring civilization, you have to like have this particular value system and set of ambitions is like, I don't know, it's very limiting in a way that limiting and depressing, you know. I think that and personally, like, you know, I would find it so thrilling and so exciting to find pond scum, you know, on another world. Something that looks like, you know, like what comes out of the drain when I snake it after like a month, you know. I would be so thrilled and excited and fascinated to find that and study that and understand that, rather than something that's just like us, you know. That's what captures like my imagination.
All good points, guys. All good points. There was a related question to that, and that was, oh goodness. No, I can't find it. I would love to know your thoughts about viruses being characterized as life, as some individuals like David Baltimore have suggested.
Yeah, I don't mind it. Short answer. As I was sort of musing about at the beginning of my talk, the exercise of trying to come up with a complete, but also like definitive definition for life, a complete definition for life is in some ways it's a useful exercise. If you're trying to ask, what are we looking for when we go to another world, or we look back in the geologic record of our own planet and we try to figure out, was life there? What was it doing? What was it like? What was the world like that it inhabited? Like, it's important to like ask, okay, what are we looking for in the first place? But there also comes a point where figuring out what does and doesn't count is not so important in like in defining the search. If we were to find something like a virus on another planet, say we figure out how to access the theorized subsurface liquid containing aquifers of Mars, we punch a straw down there and we find genetic information inside of a capsule made of proteins that genetic information specifically like encodes for. That'd be sick, you know, that'd be super interesting. And And of course, also have wider implications because a virus, sort of by definition, has to, in order to perpetuate itself, has to be able to hijack and use the cellular machinery for transcription, translation, and assembly of the cells that it infects. And so it would imply, if you find a virus, that, oh man, like that means that maybe we keep searching, we find the thing that this virus needs in order to replicate itself. But even if we didn't find that, it's perhaps, you know, possible for a virus to live independently if it's able to, perhaps if it carries around enough RNA, which is a very versatile molecule, behaves kind of like a protein, kind of like nucleic acid for like the storage of information. Like, you know, maybe it could find a way to like self-replicate independently, and that would be cool too. For me, whether or not a virus technically qualifies as life is more a matter of, I don't want to say semantics, but like more a matter of linguistics at some point. As far as the history of life on Earth goes, I think that viruses are like a huge part of the history of life on Earth. In the modern world, we know what a huge role they play in biogeochemical cycling and the overturn of certain reservoirs of sulfur and of carbon and of nitrogen and all these things in the world's oceans and in groundwater and things. Like there's, I forget the exact ratios, but for every liter of seawater you pick up, there's something like, you know, sometimes like 1,000 times or 1,000,000 times the number of viruses as there are bacteria in that liter of seawater. It's like it's truly extraordinary, just like how many viruses like exist in the world. And so they definitely play an important role in the ecology of the modern world and the history of life. Are they alive themselves? Like, it's okay if they aren't, it's okay if they are. I think that they're still an important part of like that tapestry of what it means to be alive and what it means to have a biological sphere and an abiological sphere interacting with each other. But yeah, I'm not inclined to see it so much as such a controversy.
Fair enough. Question slightly out of left field here. Dylan, what would the sky look like if your eyes could only see radio waves?
Oh, okay. So I think, first of all, you wouldn't be able to see the sky as we see it. Like it wouldn't look blue, or it wouldn't really look like anything, because the reason the sky looks blue is that sunlight comes in and it scatters off the atmosphere and it gets, redirected in a different direction towards you, right? Whereas let's say that it was just radio waves from the sun, it would just pass right through our atmosphere and it wouldn't care that the atmosphere is there. And so it wouldn't scatter back at us. So the sky would look a lot darker, I would say, first of all. But you would see lots and lots of points in the sky. And rather than being stars, those points will actually be black holes at the centers of other galaxies. Those are actually the dominant sources of radio light in the sky. And so some of these black holes will have large lobes coming off of them. They'll be like essentially a bright dot and then big blobs right around them. And those are due to jets that are being launched right out of those black holes and impacting the gas around the galaxies. And so those are called radio galaxies. The sky will sort of flicker sometimes, but it'll flicker sort of mostly on, at least as far as we know, mostly on either millisecond timescales, which is like these fast radio bursts that are coming in, which actually are like incredibly bright. Like if you're able to take high-speed video of the sky, those fast radio bursts for like the few milliseconds that they're on will be like outshine most things in the sky. But on much longer timescales and the things that I study, you'll see like supernovae turn on and gradually get brighter and brighter and brighter, and then start fading over like years timescales. So yeah, that's kind of what the sky would look like, depending on how sensitive your eyes are at radio wavelengths.
Cool. It's cool to think about what the sky looks like in different wavelengths. I know there are a bunch of different maps that people have done in using different telescopes and different instruments, like what the sky looks like in the radio or the X-ray or the infrared or the visible. There was just we talked about it last time, the new image, all sky X-ray image that was released by eRosita. And it's just crazy, the different astrophysical phenomena that you see that are so unlike what we're used to seeing when we look up in the sky, like, oh, stars, galaxies. It's like, no, now you see accretion disks around supermassive black holes, and you see, accreting binary stars and weird stuff like that. So very cool. There were a couple of related questions that I thought would be appropriate for our life and solar system people. Is there any chance of life in the atmosphere of Venus? In your opinion, will life or evidence of it be discovered on Mars? Seemed appropriate for Nicole and Cecilia, perhaps. Do you guys want to take these?
Yeah. So the first one, I don't know if I can necessarily speak to. I've done a little bit of I've studied Venus's atmosphere a little bit in the course of my coursework, but it's not something that I've directly worked with. Venus's atmosphere, presumably whoever asked the question probably knows this, so sorry if this is more information than you need, but for others' benefit, the atmosphere of Venus is very thick. The pressure is very high at the surface of Venus. It's high pressure and extremely high temperature, and so unsuitable for life as we know it. But the higher up in the atmosphere you go, the less mass there is crushing down above you, so the more hospitable perhaps Venus becomes the higher up you go. Venus also has pretty thick cloud decks of various interesting compositions that host a lot of interesting chemistry. There's likely aerosols in the atmosphere, so perhaps it'd be possible to have droplets of liquid coating the surface of aerosols that could possibly host viable cells. That said, the composition of those clouds, the composition of the atmosphere, is also really important. It's not just being at a hospitable temperature and pressure. It's also what kinds of organic and inorganic chemistries is your hypothetical cell going to be subject to. Venus's atmosphere, it has sulfuric acid clouds. It's very difficult for us, in fact, to send non-living things through Venus's atmosphere and down to the surface of Venus to collect data on to just collect any kind of data and beam it successfully back to Earth without the instrumentation degrading very, very quickly. So it seems to me that whatever cell possibly was able to make it work in some thin film of water on a mote of dust high up in Venus's atmosphere would also have to be made of something particularly recalcitrant and unlikely to dissolve, even in unlikely to be picked apart by hydrogen, free hydrogen in a highly acidic environment. This is something that exists on Earth, things that are very acid resistant. In fact, micropaleontologists take advantage of this by having in studying these ancient microbial fossils called acritarchs. They're made of this particularly recalcitrant organic material that when you dissolve away the rest of the rock with pretty concentrated acid, they are left behind as this organic residue that then you can mount and image and look at like the structure of this original cell wall. So perhaps something like that could survive there. How it would have gotten there, how it might have evolved there over the course of Venus's history is hard to say. But yeah, there's a lot of barriers, but it seems to me that they're not necessarily insurmountable barriers, given what we know about the capacity of life for surviving challenging circumstances. The second question was about Mars. Nicole, did you want to say anything about Venus before I address Mars?
Just no, is pretty much my short answer to that. Yeah, just to Cecilia's point, even if something could live there, and it's not saying that something can't, because you have things like extremophiles that live in completely bizarre places on the Earth, they had to evolve to end up there. And it just seems very unlikely that you would have something that could stay buoyant in a very specific part of the atmosphere where it could live for long enough to actually be able to live, the precursors to that. So I think just the long and the short is probably not. And if it would, that would be really, really cool. And I don't understand biology then, but.
I mean, that's an important point as well about like, you know, the evolution of extremophiles. Like that's a big open question in geobiology is were the earliest organisms what we would consider today to be extremophiles? Did life arise in what we would think of as an inhospitable place with very little available raw materials and chemical energy to actually get started and to facilitate the construction of the kinds of cellular machinery you would need to survive a sulfuric acid cloud? That's, you know, it's hard to say. We don't have an answer to that yet. And I don't know if without, but I bet it's something that someone could try to like create a laboratory model for potentially, not of like an organism that already exists on Earth, but, you know, testing the survivability of certain essential structures under Venusian atmospheric conditions. Someone's doing that, I bet. Like we could look that up. The second question was about Mars. Cameron, could you say that?
I mean, it was just related. It was, is there any chance of life in the atmosphere of Venus? And is there any, in your opinion, will life or evidence of it be discovered on Mars? So do you predict to see life on Mars?
Yeah. I love Mars. I'm biased towards Mars. I think Mars is great. And of course, it's like one of the most extensively studied bodies other than our own planet that we've ever we've ever been able to collect data for. That said, I'm fond of telling my roommates that, well, you know, geologists on Earth spend their entire career on a few square kilometers of land or a single rock formation. whereas on Mars, it might seem like from a planetary science perspective that we have so much data. We've been on the surface for years. We've been collecting dirt. We've been zapping rocks with lasers, and we still haven't found life. And it's like, well, we've only been to even with, all of this effort, we've only been to a handful of places on the surface of Mars. And for example, it'd be weird to judge the entire history of life on Earth by what you're able to find in the Grand Canyon. Compared to all of the other places and all of the other environments that have existed throughout Earth time. So I think it's possible, but I will say, given what we do think we know about the evolution of Mars, the planet itself, the fate of its atmosphere, the longevity of liquid water at its surface in the past, I think that it's probably unlikely that we would find evidence of life at the surface. I think the subsurface, or rocks that were very recently in the subsurface that might bear some evidence of interactions with microbes in deep aquifers or something like that. I think that's probably a better, probably a more likely place to find evidence. I'm not ruling it out. But what I do know and what I am sure of is because Mars' geological history is so different from the Earth, they don't have it never had a rock cycle where the oldest rocks were getting buried and uplifted and subducted or metamorphosed. Those processes didn't happen on Mars, and to our knowledge, and certainly not for as long or as constant a period as on Earth. And what that means is that Mars is a rocky planet relatively close to where we are in the solar system, with a relatively similar early history and formation story. And it still has rocks at its surface that are as old as the planet itself, which is not something that you can say for Earth. The oldest rocks that we have on the planet Earth are only accessible in a few places on the globe, and they're heavily deformed, heavily metamorphosed. They've been altered a lot by their experience of different temperatures, pressures, and fluid chemistries. Mars definitely does have a much more complete record of the planet's earliest evolution. So I think that Mars is perhaps a very interesting place to go to try to understand what prebiotic chemistry was like. What was the world like before the advent of any kind of life form? Because in that respect, Mars and Earth probably had a lot in common. And we just don't get to see rocks that old and that pristine on our own planet. So I think that, and perhaps prebiotic chemistry, is just a sexy way of saying, like chemistry without cells. But I think that it's really essential to understand prebiotic chemistry on a rocky, ostensibly Earth-like world in order to answer questions about how life arose and why did it take the form that it did, and that sort of thing. So even though I'm not terribly optimistic for the discovery of, say, stromatolites and microfossils and the kinds of signatures of early life that we see on Earth, I'm not super optimistic for discovering that on Mars necessarily. But I know that there's a lot of interesting stuff that we could learn related to this, the trajectory of life in the universe.
Nicole, or the other members of the panel, do you guys have anything to add regarding Mars?
Yeah, I think just to echo Cecilia's point that just because a planet doesn't have life doesn't mean it's not interesting. So I think we can get a tremendous amount of information from Mars climate and understand how Mars's atmosphere became the way it is, and understand, like Cecilia was saying, just the history of our own planet. And I think that's true of all the planets in the solar system. And I mostly study gas giant planets, to be fair, and life is not something I usually think too heavily about.
Rocky planets forever.
But most of my planets that I study and the stars that I study don't have surfaces. So I can even take it as, look, we can learn about how the solar system formed from Mars. We can also learn about it from Jupiter and from Saturn. And I think that's so important because that gets us to where we are, right? Without all of the precursors to the Earth, without the Earth forming, without Jupiter forming, we don't have life. So I think we have to sort of expand our scope to, do we see intelligent life on a planet? To, did the circumstances of the planet arising sort of match with what we expect it to be? And I think that's super cool because it makes all planets, regardless of whether they can be habitable or not, very important to our understanding of life in the universe.
Yeah, good point. All right, we are over on time. Are there any last things people would like to say as we close down the panel? No.
I guess I would like to add, perhaps it's a little late now, but when I first put together this talk, I sort of assumed a certain level of like base knowledge about basic chemistry, atoms, like some of the basics of biology and stuff. And we call them basic just because they're foundational, not necessarily because they're intuitive or easy to understand necessarily. So I was going to try to keep an eye. Cameron, I don't know if if comments will be available on the recording of this video, but yeah, I'll try to keep an eye on that over like the next few days too. So if you were too shy to type in a question like, oh my God, she said something about protons in passing and I don't remember what that is, or I've not heard of that before, regardless of like age or experience level, like please, like, you know, you can either like try to like look some of that stuff up on your own, but like we're here to help and to help make these topics like a little bit more accessible. And so if you have questions, no matter how like basic or simple or complicated or dreamy you think they are, I just wanted to encourage people to either reach out to me at the email that I put up or put it in the comments on the recording later.
We monitor the comment section, and when people come up with questions, I try and respond to them or get an appropriate person from the department to respond to them. So
good.
Yeah, good, very good point, Cecilia. Thank you. Okay, well, thank you all for attending tonight. Again, our next event will be Astronomy on Tap a week from Monday, July 20th, and it's all about Mars. So we can talk more about Mars then. We will do it over beers. We will have a member of one of the instrument teams for Mars 2020 Perseverance joining us, Dr. Nina Lanza from Los Alamos, and we will have recent graduate from Caltech, Dr. Alastair Hayden, who will be showing us how to explore the surface of Mars using online free tools that you can do from your computer, which is pretty awesome. So thank you. Thank you panelists for your contributions tonight and your expert opinions on everything. And yeah, we will see you guys next time. Thanks.